right, so what is the response through the extracellular matrix? Because really what we're talking about here is we're talking about increasing passive stiffness. That's what our training is designed to do. When I showed you the work from Kubo, and I've showed you the work from Fore, where they did the plyometric training, what you saw in those studies was that what happened was you increased the passive stiffness of the joints. And so by increasing the passive stiffness of the joints, what you're doing is you're changing the extracellular matrix. Because remember, we talked about the passive stiffness being determined by your by the amount of collagen, the orientation of collagen, all of those things. And that component of it was the part that was really set up by how much of the extracellular matrix proteins there were. So what's the response of the extracellular matrix of muscle to these types of loads, to an increase in load? And so this is a study that our group has done, where basically what we do is we do what's called an overload. We get that huge muscle hypertrophy. But even though the muscle is growing really, really big in a really short period of time, what we see is we see an increase in the collagen content of the muscle, and that's here. But the interesting thing is we also see an increase in the percentage of collagen within the muscle. That means that even though muscle is hypertrophying, we actually see a greater increase in collagen protein than we do in muscle proteins. And so you can see that here at the gene level. The collagen 1 gene is going up about 20-fold. Collagen 3 is going up about 20-fold. Collagen 5, which is one of the force transfer collagens in the extracellular matrix of muscle, goes up over 100-fold. So these are huge changes in the extracellular matrix. And we've talked about how the extracellular matrix is important in muscle function. This is a study that still hasn't been published. It's from a French-German colleague of ours who basically took and did the exact same experiment we just did, showing where we increase collagen content in the muscle with overload. But what they added is this drug, halofunganone. And halofunganone is a drug that is an anti-fibrotic drug. So it prevents the increase in collagen. So when they gave the halofunganone, there's the control, and what you can see is the overload, there's no difference in how much the muscle fiber hypertrophies. If anything, the halofunganone muscle fibers are hypertrophying more than the control muscles. So it looks like there might be a little bit more when you decrease collagen content. Here's the collagen confirmation of the fact that the collagen, the antifibrotic, worked. There's no increase in collagen content in the halofunganone, but there is an increase in collagen content in the control animals. Here's the important part. When we look at the isometric force, or the strength of the muscle, this is the control situation. You're going up and you're increasing force across that muscle by 3-fold. Now what happens when you give the halofunganone? You prevent the increase in collagen, and now it's only going up about 100%. So basically what it's doing is it's doubling instead of going up 3-fold. So you get about 50% of the increase in force is because of changes that are happening in the extracellular matrix. And so what that's telling us is that even though we're increasing fiber diameter to the same degree, if not more, we're not getting as much strength from the muscle. So that means that the collagen that we are making is making the muscle stronger, and it's making the muscle more robust. And for us, in this module, what we're looking to do is we're looking to increase passive stiffness and increase movement economy. So when we increase the matrix proteins, we're going to increase passive stiffness and the economy of movement. So how is our muscle transducing this shift in load across the muscle into a signal that's going to give us an increase in these extracellular matrix proteins? Well, one of the things that we had discovered a while back, almost 20 years ago, is that one of the highly increased genes that we saw following resistance exercise was this gene EGR1 and its homologue EGR2. And there's early growth response gene 1 and 2. And what we find is that within an hour and a half, the early growth response gene goes up about 130-fold. It's a huge increase in any gene. And it's the biggest increase in any gene that we've ever seen following resistance exercise. And it stays elevated. And even though 18 hours, it looks like it's really low here, it's still about tenfold higher than it is at baseline. Okay, it's back down to normal by 36 hours. And if we look at the EGR1 protein, you see that the protein goes up significantly as well, and it stays up again for more than 18 hours. Now here's the nice thing is, look at the collagen proteins here, collagen 1, 3, and 5. You can see that their mRNAs are starting to increase around the time when the peak of EGR1 is. So it looks like EGR1 is peaking here. You're making the protein, it's coming back into the nucleus, and now it's working as a transcription factor. We know it's a transcription factor for collagen 1. And so when it comes back in and you see it working as a transcription factor for collagen 1, what you can see is that you see this increase in collagen protein or collagen messenger RNAs, suggesting that the increase in collagen mRNA could be driven by an increase in EGR1. So we tested this directly by electroporating muscle. So basically, we can take any gene that we want. We can put that gene into muscle. We can give the muscle a zap, and the muscle cells will take it up. Here we gave the red fluorescence protein, and so you can see lots of different red fibers. So when we put in EGR1, and then we come back and we take the muscles a week later, you can see that the increase in EGR1 that we get at the messenger RNA level correlates really well with the increase in collagen that we get in the same muscle. So you can see that collagen 5 mRNA, for example, goes up almost completely linearly with the increase in EGR1, suggesting that EGR1 is sufficient to increase collagen synthesis. And what we now need to do is we now need to see whether it's in and of itself, whether it's required for the increase in collagen gene expression. All right, but what this suggests is that when we do resistance exercise, or when we do any type of plyometric exercise, any fast movement, what we get is we get an increase in EGR1, and that increase in EGR1 is contributing to an increase in collagen protein. So then the next question is, why is EGR1 going up? Well, EGR1 seems to go up as a result of an increase in the activity of a kinase called the MAP kinase, or phospho-ERK, or so the ERK is the extracellular regulated protein kinase, ERK. And ERK is one of the kind of canonical kinases that signals when changes in the extracellular space or extracellular matrix occur. And so here we do a simple experiment where we activate ERK using a drug. You can see it turns on and off quickly. That's what ERK does. It responds very quickly and shuts down. And that's very similar to what tendon cells do or connective tissue cells do. And so you see this increase in ERK and decrease in ERK. Then you see EGR1. So the increase in ERK peaks at 15 minutes. EGR1 mRNA peaks at 30 minutes. And EGR1 protein peaks at 60 minutes. And so now what we've got is we've got this idea that when we do high load exercises, plyometric exercises, any kind of high force exercise that's a rapid movement, we get an increase in ERK that's known from other people in the field. That increase in ERK is going to drive an increase in EGR1. When we increase EGR1 mRNA, we increase its protein. And when we increase its protein, that's enough to start increasing its messenger RNA, the collagen messenger RNA, as well as collagen protein. So now what we've got is we've got a secondary pathway here. We're doing resistance exercises, giving us mTOR activation in the muscle, but all of those fast movements are also activating ERK. ERK is activating EGR1, and EGR1 is increasing these matrix proteins.